Dear friends, today we will start with the industrial importance of proteins. What are the industrial importance of proteins? First, we started with there are a large number of industrial properties of protein. As we know that we are taking various forms of protein in our life. Generally, the people are eating for taking day RDA, recommended dietary allowance of the protein, that is in 70 grams. Okay, so to meet this requirement, we are taking as a food material. Then there are various richest sources of the protein which having a highest industrial properties. In food industries, they are utilizing in various food for making various product like as protein isolate. Okay, and that protein isolate can be used for various product too. Now first started with the industrial importance of the proteins. Gelatin. Why gelatin is used? Where gelatin is used? It is used for the manufacturing of photographic plates, animal glues, capsules. Okay, so these are the various sources through which we are utilizing. Another is casein. Casein is found in the milk protein, that is used in the various medicinal properties as well as manufacturing of various plastic base or glue. Soybean protein. Soybean is a oilseed crop, or we can say it is used in both. Soybean having the highest protein among various oilseeds. So for the manufacturing of artificial wool, fur, silk and leather, it requires. So soybean protein is the richest source for that. Another industries, medical industries or medical field. We are utilizing a plasma protein for the treatment of various serious injury, shocks or in various operation. In today's era, you will find a disease of COVID-19 that also having a treatment. There is no treatment in the world for the serious disease like viruses. So the traditional methods for curing this disease to isolate plasma proteins. Okay, same thing is single cell proteins, that is also used or produced from the hydrocarbon waste, and that can be used for animal feeding. Likewise, in many food programs where the malnutrition is going on or malnutrition is there, where the World Food Programs is based on this protein. So that much importance of this industrial importance as well as the world importance in supplementary with the diet to nourish the child in underdeveloped countries, that is run by certain international agencies. Now the seventh, or as per these laundry and detergents, these industries are utilizing certain enzymes which are having a amylase, lipase and proteases. That can be used for making a detergent or laundry. Okay, this is all about the industrial importance of the proteins. Proteins having a large number of functions. You cannot write all the functions, but you have to list out certain industries and their uses. Okay, now we will start with the metabolism, how the general metabolism of protein is started in. So as we know that there are a metabolism word include two. One is anabolism, another is catabolism. Ana means synthesis, okay, and cata means degradation, okay, or lysis. So in glycolysis we have one precursor, that is started material is glucose. Glucose is converted into pyruvic acid with the metabolism of glycolysis, and this pyruvic acid can be utilized for formation of various amino acids through transaminase reaction. Now this pyruvic acid that can also be entered into a TCA cycle as a acetyl-CoA, one of the intermediates in the metabolism. So this pyruvate convert into acetyl-CoA and TCA make a TCA cycle intermediate. Now these TCA cycle intermediates will start with the alpha ketoglutarate, and these alpha ketoglutarate can be a source for the formation of vitamins like as glutamic acid, glutamine and proline that is used in the synthesis of proteins. Likewise, oxaloacetate family that having also different kinds of reactions, and that will convert into aspartic acid, asparagine, lysine, methionine and isoleucine. These are the oxaloacetate family that also be used for the synthesis of protein. So this is the metabolism of the proteins through which the certain intermediates of TCA cycles and glycolysis can be utilized for the formation of proteins, and thereby in plants as well as in human beings that is made. Now we will see the catabolism of proteins. How the keta means degradation, that we will start with that carbohydrate moieties and enter into amino acids. Now the catabolism means same, that product has to be hydrolyzed or lysis. So the protein is hydrolyzed with the water, and that gives you a proteoses and peptones that already be taught to you in the previous lecture. The enzyme requires for that is gastric juice or gastric pepsin enzymes. Similarly, these protein further hydrolyze with the proteoses or peptones that will convert into a peptides. These small peptides are found in the intestinal trypsin juice. Now these peptides, and you eat up water, that will convert into amino acid. These amino acid can be utilized or used for absorbing in the intestine, and that deamination reaction that will convert into alpha ketoglutarate or alpha acids, and these also certain in mitochondria when it enters the TCA cycle operates, and that will convert into CO2 plus H2O. Thereby you get energy. So this is all about the metabolism and catabolism, general catabolism and general metabolism of proteins. So in examination you have to write these reactions and certain examples. How this protein is formed, if we see in any living organism, there are a triangle that is called as a central dogma. Central dogma started with the DNA to RNA, RNA to protein. DNA to RNA, that is transcription, and RNA to protein, that is translation. How it is happened, in where it is. So that is found in the ribosome, endoplasmic reticulum. These amino acids are joined together by the peptide bond, and following the various sequence dictated by the DNA, this triplet code will synthesize one amino acids, and these amino acids decide a continuous that coding sequence will decide the triplet codes of this coding will decide a amino acid. This peptide would forms a chain called polypeptides, and transcription in the nucleic acids. Transcription in the nucleic acid, DNA code for mRNA, and mRNA that will decide or translate into proteins or amino acid by the reaction called translations. This is all about the ribosome, how it is synthesized and what reaction is happening. Now we will see a certain pathways through which the various amino acids are formed. For example, first is cysteine. Okay. How the serine is formed, how the glycine is formed, and what are the intermediates. Here you will find a glycolysis, TCA cycle, and certain transamination reactions. So tryptophan, tyrosine, and phenylalanine that is produced through the phosphoenol pyruvate and dihydroxyacetone through the cyclic acid pathways. Similarly, 3-phosphoglycerate or 3-PGA from the glycolysis that will give you a serine family as well as cysteine and glycine. Similarly, in end product of the pyruvate glycolysis, that pyruvate gives you a 2 amino acid that is valine and leucine. Same thing is with that this aspartate family, that how it is formed, that is given here. So you have to write what are the moieties and what are the precursor through which the various 20 amino acids are formed. In TCA cycle, every having various precursor for the formation of amino acid. For example, alpha ketoglutarate that will gives you a glutamate, and this glutamate is also synthesized in many animals and certain birds too. This ornithine is converted into aspargine, arginine, and likewise glutamate is converted into a histine or glutamine. This is all about how the amino acid react with and what are their precursors. Okay. Now, this is also a pathways through which you can see various intermediate metabolisms. For example, Bogart Pathway is also given, then many aspartate or alpha ketoglutarate. These are the structurally which enzymes are involved in that and what the reaction is that. For example, if you see the serine, oxaloacetate, CH2COOH and C double bond O and CO, these oxaloacetate convert into aspartate, and this aspargine will convert into a aspartate convert into aspargines, through which the ATP is required and AMP plus PPI is that. Okay, so these are the enzyme, transferase enzyme, transamination and reaction are given here. Okay, so you have to remember the Bogart and Bogart pathways through which how the glutamate, alpha ketoglutarate and aspartate and oxaloacetate reaction is there, how NADP convert into NADPH. So you have to remember like this. Okay, I am giving you 3 reactions through which you can see. Okay, this is all about the reaction. I have not explained too much in the. Now what are the agriculture importance, that I will show you later on. Until that, you enjoy. Thank you.